Hi, friends. It's Mr. Rogers here, and we are going to be going over the beginnings of mitosis. And I want to try to keep it to about 10, 15 minutes. I don't want it to go too long. There's also the fill-in-the-blank notes you can follow along on, but there'll also be some drawing today notes, so we'll kind of go back and forth, and we'll see how far we get. I'm not quite sure. Kind of depends. I've never done this virtually before, so we'll see. So the first thing right away is the cell cycle is made up of 5 phases. And we've talked about the cell cycle. You've drawn it. And realistically, what you guys talked about last week was the G1, S, and G2, which are all components of interphase. They're all components of interphase. The prophase, metaphase, anaphase, and telophase are all parts of mitosis. And so when we actually talk about the phases, it has to end in the word phase, obviously. But interphase is everything that's not mitosis. It's the intermission between mitoses in the cell. That's where the prefix inter comes from. And so it's an interphase between these mitosis phases. So we go interphase, prophase, metaphase, anaphase, telophase. The order, some people just remember IPMAT. We'll kind of keep practicing over and over and over again, and we'll try to get somewhere with it. Okay. So the next stop here is just kind of making sure we understand what interphase is. So interphase is technically not part of mitosis. It's a resting phase. It's what we talked about last week. It's the, as you see at the bottom, it's the G1, S, and G2. Those are the 3 stages of interphase. It's designed to basically be where a cell lives its life. And then when it starts getting ready for mitosis, it doubles its DNA, makes copies of its DNA, and it doubles its organelles because once it splits in mitosis, we're going to go from 1 cell to 2 cell. Each cell is going to need the right amount of organelles. And so if a normal cell has 10 mitochondria, before we split, we need to make sure we have 20. That way each one gets 10, if that makes sense. Okay? So that's interphase. We're going to skip some things here. So now we're going to go into mitosis. And this is the big part. This is where we kind of been building to. This is where if you do too many mitoses, you get cancer. And so mitosis at the basic level produces 2 cells, and they're called daughter cells, and they're from one parent. And each daughter cell is identical. Identical. And that's huge. They're making identical cells. These are clone cells. They are identical to each other. That is the huge part that we need to pull out of this, okay? Because later on in this class, you're going to make cells that are not identical. And that's a big change, a big component, okay? Now, sorry, my phone keeps blowing up here. Most of these cells are body cells. So they're basically everything in your body that makes up all your cells. So you got like brain cells, you got all these other things. All the cells in our body. Then, in itself too, we are looking at mitosis because this is also how bacteria cells reproduce. This is how cells that are living out in nature that are single cell organisms, how they do asexual reproduction. So without sexual reproduction, how they do by themselves, and they make copies of themselves. Mitosis is basically the exact same process. So obviously we kind of focus on humans because we are humans. And so we call it mitosis, and we're talking about our cells inside of our body making new cells, but also understand that this is how a bacteria on the desk in front of you reproduces. So this occurs for a couple different reasons. One, like I just kind of talked about down at the bottom, single cell organisms so they can reproduce. It helps get rid of old and damaged organelles old and damaged cells that need to be continually replaced throughout the life of an organism. Or this can be used to grow. So at one point you were 1 cell and now you're no longer 1 cell. You're many cells. And so you've grown from that number because of mitosis. The nucleus of the cell is going to divide in mitosis. And so that's going to be one of the major things that's going to occur. So this is probably not in your notes. I just want to skip forward to our notes. All right, here we go. Prophase. So prophase is when the nuclear membrane disappears. We're going to draw this in a second. We're going to create spindle fibers. Now, spindle fibers come from centrioles. Centrioles was an organelle that we learned back in the day. We talked about organelles and cells. But spindle fibers are going to form up, and they're going to be used to pull chromosomes apart. Chromosomes coil up and become short and fat, and so you're going to see DNA actually form up into something that's actually visible, and this takes about an hour. So let's kind of look at my setup here. So I want to pull this up, and it's of course blurry because why wouldn't that be? Perfect. And we're going to start with interphase. So I know we just talked about prophase, and I don't know if you have a circle on your thing, but let's, on the corner, let's just draw a quick interphase. At the end of interphase, the cell has a nucleus in the middle, and the DNA is all spaghetti-like, just a massive spaghetti, okay? And that's all we really have to draw. I just want you guys to have a baseline of what a cell looks like. For prophase, what you're going to see is what we kind of just talked about. The first thing was the nucleus was going to start breaking down. So the nuclear membrane is going to break down, and the chromosomes are going to start forming, and they're going to kind of look like little X's. Instead of being spaghetti. So they actually form up into actually organized units. They're no longer just spaghetti, they're organized units, chromosomes. We also have spindle fibers forming. I said those are coming out the centrioles. This is the centriole. And the spindle fibers are starting to get made. And we'll have some over here too. Same idea, centriole, and these are the spindle fibers. And so we're starting the process of getting everything ready to split. Okay? Awesome. So let's go back here. All right. This is another picture here. Kind of looks like mine. They have their spindle fibers joining together, which isn't a big deal. You can do it either way in prophase. And these are some cells in actual life that are prophase. So if you look here, you can see this is a plant cell. This is a cell wall. You can see inside the nucleus. You can't really see, but you can kind of see the chromosomes forming up. It's kind of harder to see. This guy, it's hard to see too. It's hard to see with the basic microscopes that we have. But this is the cell membrane in here, okay? Metaphase. So metaphase, we are going to be lining up the chromosomes that we just created in the middle of the cell. So the nucleus is completely gone, and these chromosomes are lining up right along the equator, right in the middle of the cell. And I'll show you what that looks like in a second. The spindle fiber is attached to these chromosomes in the middle. And as you saw, they kind of look like X's. They're going to attach to the middle, and in further phases in the forward, they're going to break them in half. They're going to break them right down the middle, right at the centromere point. Centromere is one of these two chromosomes. These two things are connected together. Okay, these two, or the two sides of X are connected. And this takes about 15 minutes. This is relatively quick. And so, going back to the camera. Oops, sorry. So we got metaphase. And we're going to have spindle fibers. Got the centrioles on both sides. Doesn't matter if we make the equator this way or this way, by the way. You could draw it either way. It doesn't really matter. So these spindle fibers coming up. And they're going to be connecting to these X's. That was probably the worst X guy I've ever drawn. I kind of like They're like bubble letter H's to me, not X's, but everybody draws them a little bit different. Okay, so these spindle fibers are connecting on both sides. Kind of looks like a weird basketball or like an athletic ball of some kind. But you can see now they're connected to the middle here. These are the centromeres of the chromosomes. Okay? And all metaphase is really meeting in the middle. So I always like to go middle here. And I forgot to do this for prophase. Another way to look at it is it's preparing. Prepare. Cool. So that's metaphase of a cell. Next one, I guess we're going to have to actually, I always forget I have pictures too. I just like drawing things. Drawing things is always a little bit better for me. And it kind of looks just like how I kind of had it. And that's what it looks like in real life. So I think the plant one looks like an either evil spider or like a mustache or something here, crazy mustache. But you can kind of see the spindle fibers here. It's a little hard to see. This guy, it's even harder to see the spindle fibers, but you can see them meeting up in the middle of the cell here. Next one is anaphase. So anaphase, the chromosomes are going to be dividing. They're going to be splitting up. So those X's are going to go into different sides. Okay? And they're going to form chromatids, and half of each chromatid is going to go to either side of the pole. So we're moving away from each other. So you're going to notice this in the picture. It makes a lot more sense in the picture. But these X's are being pulled apart slowly. This takes about 10 minutes. So I'm going to stop here. I'm going to have another video. I know it's been about 10 minutes or so. I got a call from my wife, so I better answer. So I'll make a quick little 5-minute video added onto this, but just hold on and click the other YouTube in a second here, okay?